Good morning, good afternoon, good evening, wherever you are, whatever time it is. I'm going to adjust my screen here. Hello, hello. This is Mike Jones, Professor Mike, Professor Jones, Mike, however you wish to refer to me. This is Grand Rapids Community College, Biology 121, Anatomy and Physiology, first semester. This is the second lecture that has to do with Chapter 4. Chapter 4 has to do with all the cellular components, cellular constituents, and how cells work. So we're going to finish up Chapter 4 with this lecture. And there's a variety of odds and ends here. And toward the end of it, you'll see that we're going to go through mitosis again, but I'm going to go super fast because you've already been through all the details of mitosis. But we're going to add a little bit more to how cells function, and then when they don't function correctly or well, what happens. So as you can see on the screen, this is for chapter 4.6D through 4.10. Let me start the slideshow bit on this from the current slide. There we are, full screen. Yes. All right, let's move into it. You know I have to have my laser pointer so I can laser point at things. All right, something I didn't talk about cells, right? We talked about individual cells, and we talked about how cells differentiate from a stem cell into a specific cell that does a specific task for the body. And some of those specific tasks obviously require other cells that are connected to these cells. And so when cells are connected to each other, we call those membrane junctions. It's where the membrane of one cell will attach to a membrane of another cell. But there are different ways that they can attach depending on what these groups of cells have to do. All right, so we have three ways that cells can connect to each other, and these three ways reflect the physiology behind what these cells are required to do. So the first, we have tight junctions. These are rows of proteins that link cells together, link the cell membranes of one cell to another cell. These kinds of junctions are called tight because they are tight. They prevent the passage of substances between the cells. So wherever these substances are that want to try to weasel their way in between cells, they can't do it. Tight junctions stop them. So whatever material that is has to go through the cell. These are extremely important in the digestive system. There are other places too, yes. But the digestive system, you do not want nutrients wiggling their way between cells. You want them to go through the cells so that the cells can decide where this nutrient should go. And when you get to the digestive system, you'll see why that's so important. So these are considered to be impenetrable, tight junctions. Nothing goes between the cell. It has to go through a cell. All right. The second type, desmosomes. One of my favorite words in physiology, desmosome. And my next favorite word is the second one on the screen right down here, hemidesmosomes. That's fun to say. These are spot welds. These help to hold cells together, but they don't necessarily stop things from going in between cells. So they add a structural support. The epidermis of your skin is held together with these spot welds. And that's why you can take your skin and you can move it around. There are other reasons too, but you can move your skin all over the place and your cells don't just like disintegrate and fall apart. These desmosomes hold those cells together strongly, but not so strongly that they don't allow things to go in between the cell layers, or in between cell membranes, I should say. Next up, the third membrane junction is called a gap junction. A gap junction is a tunnel between two cells. It is a place where cells are adherent to each other, but this is a tunnel, so things can pass from one cell to another cell. And in fact, you want things to pass from one cell to another cell when gap junctions are in use. So cardiac muscles are a classic place where gap junctions are found, because you want cardiac muscle to contract I was going to say you want all those muscle cells to contract simultaneously. That's not literally true, and you're going to find that out when you get to the cardiac muscle function. But you want them to contract in a rhythmic fashion, in a sequence. And that requires the passage of certain ions from one cell to the next cell, and gap junctions allow that. Gap junctions are the only junction, let me emphasize this, gap junctions are the only junction that allow transfer of substances, ions typically, from one cell to another cell. Tight junctions do not allow that. Desmosomes do not allow that. All right, I want you to understand that how the anatomy of these junctions relates to the physiology of the cells that have these junctions. All right, so we can move on. Sure we can. I thought we could. There we go. Here's a picture, right? Descriptions are fine. They're wonderful, but pictures are worth 1,000 words. So here is a tight junction. You got it's almost like a series of staples, if you will. You know, I mean, obviously that doesn't functionally work. You don't really have staples, but they're molecular staples in a way. They staple together the cell membranes. They do not allow anything to get in between one cell and another cell. Now, unfortunately, this picture shows a tight junction, but right below it, it shows a desmosome, which they try to emphasize more on the second photo or the second graphic. This is a desmosome. Desmosomes will allow things to leak in between cell membranes, the membrane of one cell and the membrane of another cell. So if it were just desmosomes, you could get things to kind of sneak their way, substances, proteins, ions, whatnot, in between one cell and another cell. Tight junction, no way, can't get anything through there. The third one over here, gap junction, you can see they're actually pores. These are actually pores or channels from one cell to another cell. They allow, generally speaking, ions to travel back and forth, but other substances could too. So that's membrane junctions, all right? Quick and dirty on membrane junctions, moving on. We talked about the nucleus, of course, in the last slideshow and last lecture on chapter 4. We're going to delve a little bit more into the nucleus because you have to understand now, we're going to get into what is the function of DNA. And that's really what we're going to get into in the bulk of this talk. All right, so to remind you, the nucleus is has a cell membrane around it. Actually, it has a double cell membrane, frankly. And inside the nucleus are two big items that you really need to pay attention to. One is the nucleolus, or nucleolus, if you prefer that pronunciation, nucleolus, right? That is a very densely staining object inside the nucleus. The nucleolus is where ribosomes are made. We're going to find out in great detail what a ribosome does. A ribosome is a protein manufacturing machine, but proteins are made in the cytoplasm or the cytosol, the fluid part of the cell, but outside of the nucleus. That's an important understanding. So the things that make proteins, the machines, are made in the nucleolus and then shipped out to the cytoplasm. The other thing is chromatin, DNA, right? This is loosely organized DNA, fills up the rest of the nucleus, stays inside the nucleus. Understand that. The DNA does not exit the nucleus ever, except during mitosis. And you know, there's a specific phase where that happens. We'll get to that. Oh, you already know that. So we refer to this whole thing as the nuclear envelope. That's the cell membrane that surrounds the nucleus. This whole structure is called the nucleus. And then there are pores, DNA. There are bodyguards at the pores. The bodyguards do not let DNA out, but they do let other substances out. So here it is in words. Nuclear envelope is a double phospholipid membrane enclosing the nucleus, a double layer, not just one layer of phospholipid membrane. It's a two-layered structure. Separates the cytoplasm from the fluid within the nucleus. The fluid inside the nucleus, the fluidy part, is called nucleoplasm. Just like cytoplasm is found outside of the nucleus and the rest of the cell, nucleoplasm is the fluidy part inside the nucleus. Now, the nuclear envelope is continuous with rough endoplasmic reticulum. And you're going to see why that's an important process. As I said, nuclear pores, it's got holes in it. It's got holes in the nucleus. Things do have to get into the nucleus sometimes or out of the nucleus, and they go through pores. These are open passageways formed by proteins. They allow the passage of large molecules into and out of the nucleus if the bodyguard will allow it. Ions and water-soluble molecules will pass through. Other things will pass through as well, only if the bodyguard allows it. All right, here's the definition of the Nucleolus. It's dark staining. It's spherical. It is not membrane bound, meaning it floats around. I mean, I don't know if it literally floats all over the place, but it is not attached to the inside of the nuclear membrane. It's composed of protein and RNA. We haven't talked about RNA yet, but we will. We're going to get into RNA in a big way in just a few minutes. A nucleolus produces the small and large ribosome subunits. Ribosomes are composed of, as you can see here, a small and a large subunit, and those two things get together, and they are the machine that makes proteins. And we're going to talk about how they make proteins in just a minute. I promised to talk about nucleic acids, and so I will deliver. Nucleic acids. Now, you've heard of nucleic acids, I'm sure. Right? You've heard of DNA for sure. And I'm sure you've heard of mRNA recently. It's been all over the news. We're going to talk about what mRNA is in a moment. But DNA stands for deoxyribonucleic acid. Now, hardly anybody ever calls it deoxyribonucleic acid. They always call it DNA. You should understand what this word is. I pretty much guarantee you I will have a test question that will test your knowledge or test your understanding of this word. I'm going to give you other words, but DNA means deoxyribonucleic acid, DNA. DNA is the genetic material. What is genetic material? Genetic material stores instructions on how to produce proteins. So everything in your body, everything that happens in your body has to happen through a protein. A protein has to form, say the cytoskeleton of your cells. It has to form the main structural protein of your body. That's called collagen. Collagen is a protein, and that protein is defined. The ability to make that protein is defined in the DNA. Every protein in your body. And so what do proteins do? They are enzymes. You know what enzymes are now. You've studied enzymes, or you understand the definition of enzymes. All the proteins in your blood, and there are tons of them. The structural proteins of muscle and connective tissue, I just mentioned one, collagen, and actin and myosin in muscle. We're going to learn about that in a couple of units from now. Antibodies. Man, you've heard all about antibodies for a couple of years now, how important antibodies are. They're a protein. And the cell membrane components. These are all the workhorses of cells. Proteins do the work of living, but they are defined by the DNA, and the instructions on how to make these proteins are held within the DNA. And that's what we mean by genetic material. So, some definitions. You need to have some definitions. Right? Genetic information. This is the instructions to tell cells how to construct a protein, or any protein. This is stored in the sequence of DNA. Now, you don't know yet how it is stored in the sequence of DNA, but we're going to talk about that. You're going to see how that works. The next definition you need to know is a gene. A gene is a specific segment of DNA that has the genetic information in it to make one protein, or maybe even one subunit of one protein. That's called a gene. You have tens of thousands of genes in your DNA sequence. Tens of thousands. Because you have tens of thousands of proteins that have to do very, very specific jobs in your cell to make you live and to make you adapt. Adapt to when the temperature is hot, when the temperature is cold, when you start to think about something, when you breathe, when you have to filter your blood through your kidneys, whatever. There are proteins that direct that activity. Those proteins are encoded. It's almost like computer memory in a sense, or really more like a hard drive that keeps the set of information that tells the cell how to make a particular protein at a particular time to do a particular job. All right, the whole set of all of the genetic information that is contained within the DNA is called a genome. A genome. That's all of it. All of the genetic information that you have. Every single cell in your body, with a few exceptions. Red blood cells are one of those exceptions. They don't have a nucleus. But the genome is a complete set of genetic information. All of the proteins you can make, and when to make them, and how to make them, and all these other instructions are contained inside the genome. And then finally, gene expression. Gene expression is you don't want to make all of these proteins in every cell all the time. You want to make some of these proteins some of the time when you need the protein. So I'll give you an example. Insulin is a protein. It is a protein made by the pancreas. I've used this example before, but now for genetic information, we're going to use it here. You don't always need insulin to be present in your body, or at least you don't need it in high levels. Sometimes you do, sometimes you don't. When you need insulin, you want to be able to make insulin. And so you want to control when you make insulin. Well, where are the instructions to make insulin? They're in the gene. The genes of your DNA have the instructions on how to make insulin. When you need insulin, that gene we like to say that gene is turned on. When that gene is turned on, then you make the protein called insulin. And when you don't need the protein called insulin, you turn that gene off. That is called gene expression. Gene expression, it means turn the gene on, turn the gene off. You express the gene, you make insulin. If you don't express the gene, you damp it down, you don't need insulin, you don't want to make insulin. It's a waste of effort. It's a waste of energy when you're making something that you don't need to make. Now, DNA, you probably heard this thing called the double helix. The double helix. I think a good way to envision this, or to imagine what a double helix is imagine a ladder. You just take a ladder and it's got supports on either side of the ladder, and then it has rungs that go from one support to the other support. You've all seen ladders. Now, if you were to take that ladder, if it were a flexible ladder, and you could twist that ladder, if you could twist it, I'm trying to get on the screen, if you could take the ladder and twist it, then you would get this spiraling kind of ladder, a spiral staircase, if you will. That is a double helix, right? So here's a support of the ladder, and here's the other support on the other side of the ladder, and here are the rungs in between those supports. And if we twisted it in a particular way, we're going to get this twisty structure, this twisted, windy structure. That's called a double helix. The support structure of the ladder are composed of some molecules. We'll talk about those. The rungs of the ladder are composed of other kinds of molecules. These molecules that constitute the rungs of the ladder are called bases. All right, so this is what we're talking about. When we say bases, We are talking about the rungs of the ladder. Now those rungs are composed of two matched or paired, specifically paired bases. One base is found on one side of the rung. The other base is found on the other side of the rung. And so they connect together in a very specific way to form the rungs. Now, we're going to talk about the backbone in a minute. The backbone is considered the support structure, this stuff, that side and that side. And we'll see pictures in a moment that'll make it a little easier to understand. So these bases pair, and I said there's a base on one side, so like half a rung, if you want to imagine it, half a rung on one side, pairs up with a very specific base on the other half of the rung. These base pairs pair with each other in a very specific way, and here's how to remember that. This is referred to as complementary base pairing. Right? They complement each other. Oh, hello, base. Oh, good to see you. Oh, I'm so glad to see you as well. See, they're very complementary, and they pair up in a very specific way. And this is how they do. The base called adenine pairs with the base called thymine. Always. Did I emphasize that? Always. The base called cytosine pairs with a base called guanine. Always in DNA. Here's how to remember that. A little easier to remember it this way. Apples are on trees. Apples on trees, especially now in the fall around here in Michigan. Apples on trees. And the other pair, cars go in garages. Cars in garages. Cars go into a garage. Apples are on trees. A-T, C with G. Always, 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 always. So anytime you're going to see an A on one side, adenine it's called, you're going to see the base called thymine on the other side. That is the other half of the rung of the ladder. Adenine on one, thymine on the other for sure. If I come down, I see cytosine on one side, I'm going to see guanine on the other side for sure. Always. Now, this is a little bit of a DNA is a very, very long molecule. Really, really long. In fact, if you took one cell in your body, any cell, I don't care, take the DNA out of that cell, and if we were to string it out and hold it up, just the string of base pairs in your body, you have 3 billion, 3 times 10 to the 9th base pairs. If we were to string them all out like a string, and we extended it all out, it could be as long as 6 feet long, 2 meters, one cell. So that's a lot of stuff. Now it's super thin, right? It's a super thin string. We've got to wad it all up and jam it into the nucleus. How are we going to do that? There are these proteins that are called histone proteins. Histones are a particular protein that help to wind up the DNA and package it and keep it safe and packaged, but keep it accessible, except during the time of replication. When you go through mitosis, then those histones actually package it up even more tightly, and that's when we get they pack it up so tightly that they get to a thing that's called a chromosome. It is no longer a chromatin, it's a chromosome. All right, we'll get to that. All right, so we're going to back up. We're going to look at DNA, the chemical makeup of DNA. This would be one nucleotide. This is a nucleotide. This is one nucleotide. A nucleotide has 3 parts. 3 parts. We've already talked about the base. This thing over here, B, is a base. So that has to be one of those 4 bases I just talked about. Cytosine, guanine, adenine, or thymine. It must be one of those 4. That's all. That's it. That's all there is in DNA. That base is attached to a sugar molecule. Now, this sugar molecule is a 5-carbon sugar. It's called deoxyribose. Deoxyribose is the name of the sugar. And then attached to the sugar on the other side is a phosphate group. Right, so this is a phosphate group. Now, interestingly, this thing looks a lot like ATP. It's similar, has some similar structural chemical connections to ATP. But this whole thing is called a nucleotide. All right, I already told you DNA resembles a twisted ladder. Take that thing and spiral it around. You got a coiled ladder. I've already told you that the bases, this base will form a pair with another base over here. That other base would be over this direction. We're going to see that in a second. You're going to see the pictures. But that base is also attached to a sugar, and that sugar is also attached to a phosphate. So that's the other half of a rung of the ladder. We call these support structures of the ladder, we call that a sugar-phosphate chain. So the sugar and the phosphate are the thing that forms the backbone of the ladder. The bases form the rungs of the ladder. Again, here we are. Cars in garages, apples on trees. That's how you're going to see. One side is one base, the other side is the complementary base pair. And that's how we will refer to that. You have to get used to that language. A base on one side will base pair to its complementary base on the other side. Okay? Now they are attached by what are called hydrogen bonds, and we went through this in chemistry. Remember I told you that covalent bonds are the strongest bonds in physiology. Next in line are ionic bonds. They're fairly strong. And then third in line is the hydrogen bond. Hydrogen bonds are pretty weak. So you might say, well, if this nucleotide pairs up with another nucleotide over there, well, that's a weak bond. So what's the point? They're not going to stay together. Yeah, but you're forgetting one point. If I have a million bases that are paired up with their complementary bases on the other side of the ladder, I have a million hydrogen bonds. A million hydrogen bonds are pretty strong. One hydrogen bond is very weak. So hydrogen bonds are nice because you can pull them apart and you can put them back together easily. That's extremely important in how DNA works. So here it is. Here's DNA. This is one single strand of DNA, but let me tell you, DNA is almost never found as a single strand. It is almost always found as a double strand. Almost always. There are specific times when it's not. Almost always found as a double strand. And that is going to be a great distinction to the other kind of nucleic acid that we're going to talk about in a minute. So here's one strand of DNA. This is my support structure of the ladder on one side over here. Here's the other support structure of the ladder over there. Now you notice they're flipped over. These nucleotides over here are flipped over compared to these nucleotides. That's true. That's normal. It's almost like you took your ladder, you cut the rungs right down the middle, and you took one side, one half of the ladder, and turned it over, and then stuck the ladder back together. So the other side of the ladder is sort of upside down. We refer to that as an anti-parallel structure. That's the technical name for it in the DNA world, an anti-parallel structure. But still, a base here, hydrogen bond, so these little dots indicate a hydrogen bond to that base. This base hydrogen bonds with that base, this base with that base, blah, 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 blah. I haven't told you what bases they are yet, but you already know. If this is an adenine, this base over here must be a thymine. If this base is a cytosine, this base over here must be a guanine. If this is guanine, that's cytosine. If this is adenine, that's thymine. If this is thymine, that's adenine. Has to be, has to be, has to be. Always. So here we are again. Double helix. I just wanted to re-emphasize that you have the sugar phosphate structural support. They provide the support part of a ladder. The rungs are made of the bases that are base paired in a complementary fashion. A with T, apples on trees, cars and garages. Get used to this terminology. I'm telling you, that's why I put this slide up here again. You might say, oh yeah, I saw that slide already. I know you have. Complementary. And that's what it's called. You're going to get a question, I guarantee it, you're going to get a question that's going to ask you, it's going to say something along the lines of, given this sequence of DNA, tell me what the complementary base pair sequence is. And I will give you at least four examples of bases in a particular order, and you have to tell me, you have to understand complementary means apples with trees, cars and garages. The beauty of the hydrogen bond, right? So you have the sides of the ladder are supportive. That's the sugar phosphate backbone. You have the complementary base pairing, and they are hydrogen bonded in the center. And I told you a hydrogen bond is weak. This leads us to an idea of how DNA can replicate itself. If we can pull it apart, we could add new complementary bases on one side and add new complementary bases to the other side, and we would have two strands of DNA where we only had one before. That's exactly how it works. That's called DNA replication. On the point to replication, which is the point to mitosis, which I tried to beat into your heads, DNA replication, the process produces an exact copy of the DNA molecule. Not an approximate copy, exact copy. Remember, too, that occurs during interphase. DNA is replicated during interphase. Here's the steps. I told you about hydrogen bonds. They're weak. We break them apart. So we take one strand of DNA, we take the other strand, and we pull them apart from each other. We break the hydrogen bonds. The strands unwind, and they separate from each other. And now they have exposed bases. We pair those up with their complementary bases. That is under the direction of this molecule. You need to know this. The molecule that does this, that duplicates DNA, that replicates DNA, is called DNA polymerase. You are making a polymer of DNA. And then other enzymes help to connect the new sugar phosphate backbone. There are a whole bunch of other enzymes that are involved in this. But the one you really need to know is DNA polymerase. So this is a picture of how it happens. Here's our cell cycle. You already know about the cell cycle. So here in interphase, during the S phase, is when this occurs. So we take that double-stranded, we pull it apart, we have exposed bases here and here, we find complementary bases in the inside the nucleus, and those complementary bases bind with their complementary pairs, and then DNA polymerase and other enzymes stitch those together into new double-stranded DNA. That happens. All that stuff happens during interphase, during the S phase in particular. And this is all in preparation for mitosis. All right, so now how does the next question then becomes, how does DNA encode the instructions to make a protein? Now it gets a little bit maybe, I don't know, complicated, or the challenge is to take your DNA and convert it into the instructions for a protein. So there's a little bit of an understanding, kind of a gap there. I gotta have a little Coke. My throat's dry. So the genetic information is stored stores a correct sequence of amino acids. So remember, an amino acid is a molecule that when you string amino acids together, you can make a protein or a polypeptide chain. You string them together in a head to tail fashion. Okay. I have to code, the point of DNA is to be able to tell the protein making machinery, put this amino acid next, and then put this amino acid next, and then put this amino acid next, and this one, and this one, and this one. So that you have the sequence of amino acids in the right order. And that has to be coded for in the DNA. How does DNA do that? It does it by this thing called a triplet code. Now I'm going to explain to you why it's a triplet. Triplet means 3. What we mean by a triplet code is that we have 3 bases in a row. So say on one side, on one strand of DNA, so you would say maybe guanine, cytosine, and adenine, GCA. So we'll say the GCA, that 3 in a row, codes for 1 amino acid. And this took years, probably took more than a decade of researchers trying to figure out, does one base code for 1 amino acid? Well, so think about that. If there was one base on DNA, say adenine, that coded for 1 amino acid, well, you only have 4 bases. If you have 4 bases, then you could have only 4 amino acids. Well, it turns out that you have 20 amino acids. Maybe there could be 21, but you have 20 for sure, 20 amino acids that make up all the proteins of your body. So what that means is you have to have a sequence of DNA that is able to code for at least 20 amino acids. Well, if it's one base to 1 amino acid, well, that's only 4 amino acids. That's no good. That's not good enough. If you had 2 bases that coded for 1 amino acid, two in a row, then you'd say, well, okay, that allows me to get up to, I can get 16 combinations that way. Because you have 4 bases, and if you had 2 possible bases at each position, you would have 4 times 4 possibilities. So that would give you 16. You're getting close. That's pretty close. That's 16 amino acids, but you need 20. So you would be leaving 4 amino acids out. So that's not good either. So finally it was hit upon, well, if we had 3 bases in a row, that would allow us to code for 64 different amino acids. So you've got overkill there. But you have to have at least 3. So long story short, it was figured out that this is called a triplet code. So a series of 3 bases on DNA, 3 in a row, code for 1 amino acid to make a protein. The sequence of the base in a gene determines the amino acid sequence in a polypeptide. So follow that. You've got an amino acid, amino acid, amino acid, amino acid. A specific protein will have amino acids strung together in a very specific sequence. That sequence has to be coded for in the DNA. So if each one of those bases, or each one of those amino acids, is coded for by 3 bases in the DNA, every amino acid has to have 3 bases that represent that amino acid in the DNA in that order. So you have to have the 3 that represent this amino acid next to the 3 that represent this amino acid, next to the 3 that represent that amino acid, and so on and so forth, on and on and on and on. So they have to be in the right sequence in the DNA in order to make the protein in the right amino acid sequence. Understand how that works? Sequence of bases in a gene determines the amino acid sequence in the polypeptide. The DNA stores the master copy. So the master copy, the instructions on how to make any protein. So if it's insulin, the instructions are in the DNA in order. Stays in the nucleus. I've told you that it doesn't leave the nucleus. However, protein synthesis occurs in the cytoplasm. So you have to get those instructions from the nucleus, from the DNA, out of the nucleus, into the cytoplasm to direct the production of proteins. It turns out that there is another kind of nucleic acid that can do that. And that nucleic acid is called ribonucleic acid. So RNA. RNA is a kind of amino or a kind of nucleic acid that can move from the nucleus into the cytoplasm. DNA stays behind. RNA can move into the cytoplasm. That can direct the production of proteins. Let's see how that works. So RNA, now we're talking about RNA. Remember, RNA stands for ribonucleic acid. It's a slightly different sugar. The sugar is called ribose. So there's a difference between DNA and RNA. Different sugar. The phosphate is the same. So DNA and RNA are similar in that way. Same phosphate. They're different sugars. And one other huge difference is there is one different base that's used in RNA than that is not used in DNA. And that base is called uracil. Where is uracil? Here it is, uracil. So uracil replaces the base called thymine. Thymine is in DNA. Uracil is used in RNA. So those are the big differences between RNA and DNA. Another big difference is DNA is almost always found in a double-stranded form. RNA is almost never found in a double-stranded form. It's almost always single-stranded. That's another big difference. All right. We still have complementary base pairing, though. You'll see why that's necessary. I mean, I told you this is a single strand, but it's still important that it can base pair, complementary base pairing. So if you're complementary base pairing RNA to RNA, cars are still in garages. That's the same as in DNA. But apples are underground. I don't know how you're going to remember that. Apples are no longer on trees anymore. A and U instead of A and T. And you're going to see where that's a big deal in a minute. So another difference is RNA is much shorter than DNA. DNA is literally millions of base pairs long. One stretch of DNA can go on and on for millions of base pairs. RNA will only be a few hundred nucleotides long, usually. Could be a few thousand nucleotides, but it's not millions for sure, not millions. Much shorter, single-stranded usually, and you replace thymine with uracil. Okay. Now, you know the process of DNA replication. We've already been through that. That is done by DNA polymerase. Great. Now, if you have to make this RNA that can go from the nucleus to the cytoplasm, the process by which you form an RNA molecule is called transcription. So transcription takes the DNA sequence and you convert it into an RNA sequence. The RNA that is made or created during that process is called messenger RNA. That's the message. That has the message. That's why we call it messenger RNA. It has the message from the gene sequence on DNA. It takes that message in the right order and takes it out into the cytoplasm so you can make the protein from that message. So that's messenger RNA or mRNA. Now, yes, the vaccines the Pfizer vaccine and the Moderna vaccine are what are referred to as mRNA vaccines. So we'll talk a little bit about that. But they have a message. So when you're making a protein, the message comes from the DNA, is converted to mRNA, comes out of the nucleus into the cytoplasm, and directs and guides the production of the protein that DNA sequence codes for. What makes that? RNA polymerase makes messenger RNA. The DNA polymerase makes more DNA. RNA polymerase takes the DNA sequence and converts it into an RNA strand, and that's what's allowed to leave the nucleus. Okay, so RNA molecules, steps in the transcription. Remember, this process is called transcription. We're converting it from one kind of nucleic acid into a different kind of nucleic acid with a few small differences in between the two. We still haven't gotten to making a protein yet. That's the next step. RNA polymerase recognizes the correct strand of the DNA. Remember, when I say correct strand, how does it know, right? You have two strands of DNA. Well, when I pull them apart, how do I know which strand I'm supposed to go? The RNA polymerase is smart like that. It knows to grab that strand or that strand. It says, yeah, I don't care about the other strand. I want this strand. It recognizes the correct strand. There are ways that we've learned that happens. Doesn't matter for our purposes. Grabs the correct strand. RNA polymerase grabs onto it. It unwinds a section of the DNA. So remember, DNA is millions of base pairs long. If we're just going to pull apart, say, 20, 30, 40, 50 base pairs, so that the RNA polymerase can grab onto that strand and create mRNA. When I'm done making that copy for mRNA, those two strands of DNA will go back together. Complementary mRNA nucleotides pair with the DNA bases, but remember, uracil is used in the RNA. In making the RNA, uracil is used where it would normally require thymine. We're going to see how that works. There's a termination signal. So there's 3 base pairs in a row that tell the RNA polymerase, start here. And there's another 3 bases later on that say, stop. So we call those a start sequence, and we call it a stop sequence. Right? So the RNA polymerase knows, I'm supposed to go along, I'm supposed to make more mRNA, more. It gets to this specific 3 sequence code, and it says, oh, that's where I'm supposed to stop. I'm done. It lets go of DNA. mRNA leaves the nucleus through a nuclear pore and attaches to a ribosome in the cytoplasm. Remember, the ribosome is the protein-making machine, but it needs instructions, like all machines need instructions. Okay, now I have to set the stage here. This is DNA on this side. Okay, so all of this is DNA. Over here, we are transcribing this section of DNA, and we're turning it into mRNA, messenger RNA on this side. I want you to start down here at the bottom. So I have a G, right? Garages and cars. All right? That's the same in RNA and DNA. Okay, so I've got guanine. I'm going to have a cytosine over on RNA. I have a cytosine here. You already know you're going to have a guanine in RNA because those are the same in RNA and DNA. Here, guanine again. I have a cytosine over there. Great. Okay, we're good so far. We go to the next one here. Thymine. Ooh, but thymine pairs with A, right? Apples and trees. So thymine in DNA pairs with an adenine on RNA. Oh, okay, that's fine. I have adenine in RNA. Great. If I have an adenine in DNA, I would like to pair that with a thymine, right? I would say, oh, apples on trees, but then I have to remember, no. I don't have thymine in RNA. I have to change that to uracil. All right, so G with C, C with G, G with C, T with A, it's fine. A, where you would expect to see a T, in RNA, you replace it with U. This is called complementary base pairing. It's going to be very important that you understand this idea, because it's going to get more complicated as we go along. So run this through in your head several times so that you understand complementary. Now, I have to take this mRNA. So I have an mRNA. I have a single-stranded mRNA. I'm going to take it out of the nucleus into the cytoplasm. I have to turn it into a protein. That process is called translation. So I already told you each amino acid is specified by a sequence of 3 bases on DNA. That 3-base sequence is called a codon. A codon is 3 bases in a row that codes for a particular amino acid, but it has to go through this transcription and translation process to become an amino acid. But still, that 3 bases on DNA codes for a specific amino acid. That 3-base sequence is called a codon. Where protein synthesis occurs in the cytoplasm, I've said that several times. mRNA has to get out of the nucleus, has to get out to the cytoplasm, bind to a ribosome. mRNA is now the template on which the ribosome will grab onto it, and the ribosome now will interpret or translate what the mRNA sequence is, and it will turn it into a protein, an amino acid sequence. So we say the mRNA is a template. It's a template for protein synthesis. At the ribosome, the genetic code carried by mRNA now, because it's been transcribed, DNA was transcribed into mRNA, is used to synthesize the protein. The process of translation is the process of converting the genetic code, the base sequence, into an amino acid sequence. That is called translation. So transcription happens first. That happens in the nucleus. Translation happens next. That happens in the cytoplasm. The 3-base code, 3 bases in a row that code for a particular amino acid, is called a codon. So here's a picture. Now, it can be confusing. First, understand. This is my DNA, my double helix DNA. So I have a double-stranded DNA. I am inside the nucleus. You already know that. DNA strands are pulled apart by RNA polymerase. So this is one DNA strand over there. Here's the other DNA strand over here. I want to make an mRNA. So here is the sequence of mRNA. I'm creating that with RNA polymerase. Now they've blown it up a little bit so that you can see. Here's the DNA strand, has a T, A, C, 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 G, A, G. Okay? So this is what I'm going to give you on a test. A test question will be, here's the DNA strand, or I can say, here's the DNA strand that I want to convert into an mRNA. What would be its complementary mRNA sequence? Or I could say, what would be the complementary DNA sequence to this sequence of DNA? Or I could say, I have this messenger RNA sequence. What is the corresponding DNA sequence, complementary DNA sequence to that mRNA? I could say this in a number of different ways, and you have to be able to go one way or the other with that. That's the expectation. All right, so I have a DNA strand. So T, great, thymine. I have adenine in RNA. Easy. Oh, I have an A here. Well, I don't have it. I would like to say, a T, but I don't have thymine in RNA, so that's got to be a U, right? C goes with G, that's fine. C with G, that's fine. C with G, that's fine. I got a G that goes with C. I have that available in my mRNA. I have an A here. I'd like to bind that to a thymine. Not available in RNA, so it's got to be a uracil, and G and C. So that's my new sequence, my mRNA sequence. That leaves, right? The mRNA leaves out through a nuclear pore, goes out in the cytoplasm, which is where the ribosomes are. Ribosomes are just waiting around. And they want to grab on. So you have this small subunit here, ribosome. Here's a big subunit. So those two subunits, they act like a big clamp. They clamp down on the start sequence. There is a start sequence on the mRNA that the ribosome recognizes. Now, we have to convert the mRNA to an amino acid sequence. Okay, the ribosome is responsible for that, but there's one other kind of RNA that I haven't told you about yet that you have to understand. And that is this thing. This thing is called tRNA. Ugh, another kind of RNA? Yes. Yeah, that's right. tRNA stands for transfer RNA. What does it transfer? It transfers an amino acid. So on one end of a tRNA is a 3-base sequence. On the other end of the RNA is an amino acid that is attached to the tRNA and is specific for that particular sequence of RNA. This 3-base sequence is called an anticodon. But it makes sense. Here on DNA you had a codon. That was transcribed into a codon, a 3-base codon, on mRNA. So I have a codon here. Well, I have to translate it. If I'm going to translate it, I have to have an anticodon. The anticodon is on tRNA. It has a complementary base sequence to my RNA. Yes, I have at least 20 different tRNAs because I got 20 amino acids. So that tRNA with the right amino acid on it attaches to this complementary base pair sequence. The codon and the anticodon get together. I have an amino acid. Great. The ribosome then, ribosome hangs on to that, moves on down 3 more bases, and adds another tRNA with its particular amino acid to this growing chain of polypeptides. Amino acid, amino acid, amino acid, amino acid, amino acid, specific to the 3-base sequence as we go along. So yes, in the process, the tRNA binds, it gives up its amino acid to the ribosome. The ribosome says, thank you very much. I'll take that amino acid. You can go now. The tRNA will leave without its amino acid, and it will go get another amino acid just in case it's needed again. On and on and on and on and on until the ribosome gets to what's called a stop sequence. It will just dutifully add amino acids, add them. Eventually it gets down here and it finds this thing called a stop codon and it says, oh, I'm done. And it lets go of the mRNA and I have a protein. That whole process is called translation. So that part of the process is called protein synthesis. Protein synthesis means translation. Protein synthesis requires that amino acids are added to a growing polypeptide chain in a proper sequence. Extremely important. Transfer RNA is the vehicle. Transfer RNA has RNA at one end, a complementary sequence, and it carries an amino acid at the other end. tRNA binds its amino acid, so it's floating around, has an amino acid. It's looking for a ribosome with mRNA with a particular sequence. It's got to be the sequence, and it has to be a complementary sequence. Once it finds it, gives the amino acid up to the ribosome snaps on an amino acid to another amino acid to another as it keeps on going. The tRNA has the anticodon. The mRNA has the codon that was transcribed from the codon on DNA. Ribosome moves down the mRNA. Each tRNA brings its amino acid. The polypeptide chain gets longer and longer and longer. Remember, 20 kinds of amino acids. Now there are 64 possible codons. How does that happen? Because you have 4 bases in DNA, A, T, C, G. If I put 4 possible bases in one slot, and I have 4 possible bases in the next slot, and then I have 4 possible bases in the next slot. I'm running out of hands here. Right? 4 times 4 times 4 gives me 64 possible combinations of those 4 bases. That's way more than I need, but 4 is not enough. 4 times 4, 16, that's not enough. 4 times 4 times 4, 64, that's enough. It's more than enough. You have an initiation codon. It's always, on the mRNA, the initiation codon is always AUG. That is the start codon, if you will. Initiation codon. That's the one that the ribosome's looking for. D, 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 D. It sees AUG. Boom. Start here. There are three different stop codons. Doesn't matter what they are, but they're stop codons, right? They say you're done. mRNA codon codes for an amino acid. There is a corresponding tRNA anticodon. They must complement each other. So here it is again. It's another picture. Same process. Here's my mRNA. Here's my large ribosomal subunit, small ribosomal subunit. It finds AUG, it finds the start codon. It starts manufacturing polypeptide chain. Down here they're showing you, here's AUG, that's codon number 1, that codes for the amino acid called methionine. You don't have to know what codons code for what amino acid. That is not an essential part of this. If you take an advanced biochemistry class, yes, you're going to have to know that. For our purposes, I don't care. They're just telling you AUG codes for methionine. Right, the next one down, GGC codes for glycine, etc. On and on. Each codon is 3 bases long, codes for 1 amino acid. That is called a triplet code, triplet codon. So, chart form. These are the charts. You print them off, you put them under your pillow so that this knowledge can soak into your brain while you're sleeping. Transcription. Let's back it up. Let's kind of review this. Transcription occurs in the nucleus. Okay? Transcription turns DNA into messenger RNA. It is performed by an enzyme called RNA polymerase. The enzyme pulls apart and unwinds a subsection of DNA. It knows which strand to choose as to make its copy. The mRNA, or when the RNA polymerase is done, the mRNA is released. The DNA goes back to the double helix. The mRNA passes through a nuclear pore, goes into the cytoplasm. Once it's in the cytoplasm, the process of translation can occur. Ribosome binds to the mRNA at the start codon or the initiation codon. tRNA brings in, has a complementary anticodon, and it brings an amino acid with it. Those amino acids are strung together. You get peptide bonds that are formed between each amino acid. That growing chain, and it depends how long is the protein. Is the protein 10 amino acids long? Then you need 30 codons. You need 30 base pairs in a row to code for 10 amino acids. If the polypeptide or the protein is 100 amino acids long, then you need 300 triplet codons to code for that, etc. On and on. Depending on how many amino acids, multiply times 3, that's how many bases of DNA you need. So that's how you make proteins. That is called translation. This whole process is referred to as protein synthesis. We've talked about the cell cycle. That's the last part of this. I'm going to very quickly go through this. You remember, this is where the genetic material is replicated in the S phase. That's part of interphase. The first part of interphase is G1. That's where cell growth, and it does the cell all the way through this interphase. It's still being a cell. If it's a cardiac cell, it's contracting. If it's a kidney cell, it's filtering. You know, they're still doing the thing that cell is supposed to be doing. If it gets into S phase, it's going to replicate the genetic material. It's getting ready for mitosis. So we've already been through all this kind of stuff. You can review these. Remember, the process of mitosis is to create two identical cells. Where you had one cell before, you want two identical cells when you're done. Cytokinesis is when the division of the cytoplasm, right? You've put the copies of DNA in one cell and the other cell. Now you have to pinch them off. Pinch them off is called cytokinesis. This is the order: prophase, metaphase, anaphase, telophase. PMAT, PMAT. Here's what happens. We've been through that in the other lecture. Review all these, understand them. Here it is in pictures. Round and round and round you go. Cytokinesis is it's not a phase, right? See, and here it says it begins during anaphase. So cytokinesis is not a phase it's not part of the PMAT, but it is a necessary part. It begins during anaphase, continues through telophase, and eventually results in the formation of the two cells, the separation of the cell membrane of the original cell, and creates two cells. All right, now we have to talk about control of cell division. So cells have to be told when to multiply. When do we need more cells? You don't want cells just deciding on their own at whatever point that they want to multiply. You want that process to be well controlled and dictated by the forces that dictate when this should happen. So, cell division varies by cell type. You need skin cells all the time. They are constantly dividing. Intestinal cells are constantly dividing. Because as you eat something, that food stuff goes through your intestines and it starts to kind of, in a sense, scrape along the cells on the inside of your intestines, and some of those cells get pulled off and they need to be replaced. So they divide quite often. Blood-forming cells divide quite often. Neurons don't divide much at all. You don't need a whole lot more neurons. The ends of chromosomes, the very, very ends of chromosomes, are called telomeres or telomeres. Not to be confused with the T in PMAT, right? Prophase, metaphase, anaphase, telophase. Telomeres are the ends. So like telophase is the last or the end of mitosis. Telomeres are the ends of chromosomes. They act like what we call a mitotic clock, because the length of the telomeres gets shorter and shorter and shorter with each replication of a cell. And that does have to do with cellular aging. It's beyond the focus of this course. Now, cells have to divide when they're supposed to divide, at the pace and rate at which they're supposed to divide. One of the ways that they know when to divide and when not to divide, there are hormones or growth factors that will tell a cell, go ahead and divide, or no, you need to stop. There's also a thing called contact inhibition. That means if you have a bunch of cells and you're filling up an area. So you have a wound. Let's say you have a cut on your hand, right? There's a gap. There's a gap between where the, where one side of the skin and the other side of the skin, they're pulled apart because of the wound. You want the skin cells to meet each other, but you don't want them to keep growing on top of each other, right? So when they meet each other, that means they know that I have closed the wound, and that's called contact inhibition. Those cells will say, oh, I've met the other side of the cell, or the other side of the wound. I don't need to make more of myself. I'm good. Everything's fine. Tumors can result from a loss of control of either contact inhibition. So tumors don't have contact inhibition. They will continue to divide, and they will jam pile one on top of the other. So you get these piles of cells. That's not good. Cells are supposed to die. They have a programming in the DNA that tells them this is when you should grow and this is when you should stop. Contact inhibition is one of those ways. Responsiveness to hormones, responsiveness to other chemicals in the body. That's another way that cells know when they should continue to divide or when they should stop, when they need to quit growing. As we age, cells become less responsive to those hormones, less responsive to contact inhibition, and they often become less controlled. Now, that's not every cell in your body, but there are enough cells in your body that lose the ability to know when they should stop growing, or in fact when they should it's just going to sound like a weird term, but it's a process that is called apoptosis. Apoptosis is a programmed cell death. When cells are supposed to die, they need to die. They need to, and in fact, they kill themselves, which sounds a little weird, but they need to know when to grow and when not to grow and when to stop all of their functions. So on any given level, a cell can stop growing, and in fact, it can kill itself in order to maintain the function of the rest of the organ or organ system. Right, so that's this process. That's called apoptosis. Apoptosis is what we call programmed cell death. That means the cell is induced to commit suicide. It is told, you're no good. You're not helping the rest of us. You need to die. Kill yourself. It's a little bit more dramatic when we talk about it, on an emotional level or on a human level. Cells don't work like that, right? They just know, I'm no good. I've got some damage. I need to die. So they kill themselves, and that process is called apoptosis. And there it is. Apoptosis is initiated by enzymes. There are enzymes in the cell. Yes, in your own DNA. Your own DNA will tell your own cell, okay, that's it. I'm done. I got to kill myself. I got to end it. And so it will start to destroy its own DNA. It will destroy its DNA polymerase. It will digest its own DNA. It will digest the cytoskeleton. And so the whole cell really just falls apart. Now, one of the ways, this is the last thing, and I'll tell you, one of the ways in which programmed cell death works in development, when you're in utero and you are growing a hand or a foot, this is true for both hands and feet, what you do is you grow a mitten. Now you might say, well, we live in Michigan, of course we grow a mitten. No, I mean really a mitten. You have 5 bones. You'll have a fifth digit, a fourth digit, a third digit, second and first digit, and they will grow in this fashion. And then the cells in between those bones will commit suicide. They will go through a process called apoptosis. And so when this series of cells dies, you get a thumb. When this series of cells kills itself, you get a first digit. When this one, you get a second digit. When this one does, you get a third or fourth and a fifth digit. So you form a mitten, and then your cells undergo apoptosis, boom, 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 and you get a hand. That is programmed cell death. The cells in between the bones of your fingers are supposed to die. There is a process. Sometimes it doesn't happen. So some people will be born with united digits. It's fairly rare, but it's nothing bad. It's just that these cells didn't die. If they didn't die, those fingers are attached. So that's a standard way in which cells are supposed to die. They're supposed to kill themselves. They are told to kill themselves, to destroy themselves in order to make development proceed. If they don't do that, if cells don't kill themselves when they're supposed to, they can form tumors. Tumors are cells that are unresponsive to hormones. They're unresponsive to the signals that are telling those cells, you need to die, you need to stop doing what you're doing. When they don't listen to that, that's called a tumor. And that can create cancer, and that can be bad for you. So cells need to respond to these signals when they're supposed to divide, when they're not supposed to divide. And they need to listen to those signals. All right, so that's the end of this part 2 of chapter 4. That's a lot of stuff to go through. Please learn these lessons and structures, be able to understand what complementary base pairing is, understand the differences between RNA and DNA, understand the cell cycle, understand transcription and translation. Know the differences between those two things and the similarities and why that whole process has to happen. And that whole process becomes a whole concept that we call protein synthesis. Essential. Thanks. That's it for part 2 of chapter 4. We will move on to chapter 5 next. We're going to talk about tissue types. Thanks, and look forward to that.